Yeah, so hello. This is our group, which is microbes working on microbes, and this is me, Chizia Abago, Caitlin, Elijah, and Maddie.
Sorry, I'm trying to figure out the unmute button. Hi everyone, I will be introducing myself first. My name is Maddie Gomez and I go to Polytechnic School in Pasadena. I am 16 years old and a rising junior this year, and I joined the SRC program because I wanted to learn more about biology, experience lab work outside of my school science class, and learn more about the trillions of microbes functioning in our world. A fun fact about me is that I am currently reading The 48 Laws of Power by Robert Greene.
Hello.
Hello, my name is, oh, sorry. Hello, my name is Chizia Abago, and I also go to Polytechnic School, and I'm a senior. At least my one fun fact about myself is that I have two dogs, one dog named Ash and another small dog named Bolt. And my favorite book is Americana. The reason why I joined the SRC program is because I wanted to have a lot of just wanted to have lab experience, as well as just learning about microorganisms and learning about how they contribute to the environment and to like our ecosystem.
Hello, my
Hi, my
name
name is Caitlin Ong. I attend Blair High School in Pasadena, and I am in 11th grade. I joined this program because I've always been interested in science and learning how the world works, and I wanted to learn more about science in general. And a fun fact about me is that I like swimming and playing water polo.
Hello, my name is Elijah Ford. I go to Cabrillo High School in Long Beach, California. I joined this program so I can learn more about microbiology. And a fun fact about me is that I like to skate and I like to play basketball. And I have a microbe is an organism that is microscopic. It can only be seen under a microscope. Um, sorry. First, I'll be explaining what a mudwad is and how it works. Like I said, a microbe is an organism that is microscopic and it can only be seen under a microscope. Um, an anode is a conductive graphite fiber felt electrode, and a cathode is also the same. So a mudwad is made up of an LED on the right of the circuit. I mean, on the sorry, an LED. A mudwa is made up of a red light on top of a circuit board on top of the mudwa. A mudwa is a microbial fuel cell. It is a device that uses microbes found in soil to produce electrical energy. It's made up of the two electrodes, the anode and the cathode. As you can see, the cathode goes on top of the soil. The anode is placed under the wet soil, and over time, the microbes generate electricity form on the anode. The electrons travel through the wire to the cathode, causing the LED to light up, and it will continue to do that.
So now that you know like how the Medwat works, so specifically what did we do? And so what we did is that we worked with microbial organisms that are found in practically all soil sediments and streams on the planet. The mud that we specifically worked with was found in the Caltech pond, which contained a lot of methane, which I can say just like it was very like hard to take it out and extract it. And so we used like a lot of buckets. And although it smelled very bad, it was very fun to do the experiment or just like start the experiment overall. And so the mudwa applies these unique metabolisms by providing electrogenetic microbes with a certain configuration of two graphite-based electrodes placed in environments with different amounts of oxygen. So this is seen with the cathode and anode. That was what Elijah explained in the last slide. Then, given a certain experiment with a given hypothesis of our choosing, we analyzed our mudwat samples and tested our hypothesis to see ways in which these mudwats can generate bacteria and blink at certain microwatts.
So now that you know what we did, why we do it. So, the reason we conducted our experiments was to learn more about the bacteria living inside our mudwats. We were particularly interested in how bacteria thrive using electron transfer in anaerobic environments. We also wanted to see how the bacteria would react in different environments with different organic and inorganic substances and test if there were ideal conditions like an ideal temperature or amount of light for maximum growth.
Yeah, and so now at this point, you may be asking yourself, but what kinds of microbes did they work with? There are trillions of microbes in our world, but in the soil of our mudwat, we studied two different microbes, the geobacter and the Shewanella. Similar in the ways that they both have the unique ability to expel electrons to compounds outside of their bodies, the geobacter and Shewanella microbes donate their spare electrons to the anode of the mudwat through three ways. And as you remember, the anode is the graphite felt disc that is at the bottom of the mudwat with a thin layer of soil at the bottom and a lot of soil in between the anode and the cathode. The geobacter and Shewanella microbes donate their spare electrons to the anode of the mudwat through these ways: mediated transfer using electron shuttling biomolecules, nanowire transfer using conductive limbs of the microbe attached to the electrode, or direct transfer from the microbe cell wall to the anode. This means that microbes can either send electrons to the anode through smaller molecules through their arms or through direct contact with the anode. Once electrons are transferred to the anode, the mudwat process can begin. So the geobacter specifically is an anaerobic respiration bacterial species, which was found to be the first microbe able to oxidize compounds and metals. The geobacter especially likes to use iron oxide as an electron acceptor when it eats the carbon in the soil and is very conductive. The discovery that the geobacter usually doesn't need contact to transfer electrons only added to its very impressive reputation. So far, the geobacter has been used in bioremediation to eat oil-based pollutants and carbon-containing material. And just to clarify as well, anaerobic means without oxygen and aerobic means with oxygen. And then the Shewanella next, meanwhile, is a little bit different. We actually got to meet and read the scientific paper of the first person to isolate this microbe, Dr. Ken Nielsen, which is really cool. And the Shewanella is a facultative bacterium, which is a type of bacteria capable of making ATP through both anaerobic, so without oxygen, and aerobic respiration. So it can survive in both oxygen and no oxygen environments. It prefers to live in soil that is rich in heavy metals such as iron and lead, but it can also target other substances like nitrates. And the Shewanella's full name is Shewanella onidensis MR1, and it comes from the lake that it was found in, Lake Onidensis, and the fact that it is manganese reducing.
Now, on to my experiment. We are doing the experiments in order of youngest to oldest. For my first experiment, I sliced bananas and I put them un- under the cathode. I put them on top of the soil. I put 2 to 3 bananas. I sliced them. Well, it created fungus, and then my hypothesis was that the sugar from the banana would make the microwatts increase and cause my LED to blink faster, but it was inconsistent because Motowatt A produced 123 microwatts and eventually started slowing down, and Motowatt B increased in speed while Motowatt C decreased in speed. Eventually, my Mudwatts died and they stopped blinking. So I decided to do something else. And this is where experiment two comes in because I decided to put disinfectants in them. Even though they stopped blinking, I wanted to see how they would react to the disinfectants. So I sprayed all three of the Mudwatts with Clorox. And immediately they were revived and they started blinking almost every second. It was interesting because I wondered whether or not the microbes in the fuel cell started producing electricity because they were trying to survive, or if the chemicals caused an anaerobic reaction. Mono A produced 220 microwatts. Even so, after a while, they stopped blinking and all three died once again. It was interesting. For the final experiment, I decided to use different disinfectants. For A, I used Clorox. Even so, after a while, oh, sorry. For A, I used Clorox, and for C, I used Lysol. But for B, I used a teaspoon of sugar. Again, Mudwat A immediately revived after the Clorox, but the other two were stagnant. After 2 days, Mudwat C with the Lysol started blinking, and it took 5 days for Mudwat B to blink, as expected. I believe the fungi created by the bananas in experiment one interfered with the respiratory functions of the mudwats. Then the Clorox attacked the inhibitor, which was the fungi, giving the mudwats a temporary boost of energy from the backup and the electron transfer. But a disinfectant is still made for one reason, to disinfect. So over time, Clorox killed many microbes, causing them not to be able to produce electricity. For experiment three, up to this date, the MOATs have been blinking for 2 weeks.
So with my mudwats, I conducted two different experiments. So the first one was related to temperature. I placed mudwats 2 and 3 in the refrigerator to see how cold temperatures would affect it. And I left mudwat 1 alone so it could be my control. My hypothesis was that the electrons would stop flowing and the lights would stop blinking. And this was what happened. After being placed in the fridge, the microwattages of mudwatts 2 and 3 dropped, as you can see in the graph, and stopped blinking after a few days. Then I took the mudwatts out of the refrigerator to see if they could recover, and they did. It took about half a day to be brought back up to their previous microwattages. This can be explained by how particles move slower in cold temperatures. I like to think of it kind of like particles in a solid move slower than particles in a warmer state of matter like a liquid. It's kind of similar to that. The slower movement of the particles made the electron flow slower, perhaps to the point of non-existence, so the light no longer had the power it needed. For my second experiment, I connected mudwatts 2 and 3 in a circuit by changing the wires of the anode and the cathode. I predicted that the microwattages of both containers would increase. So as you can see in the graph, the microwattages of both mudwatts did increase, but not by very much, and definitely not by as much as I had expected. The increase in wattage can be explained by the fact that there are now two power sources to power the light instead of just one.
Yeah, so for my experiment, I decided to test the effect of inorganic substances such as copper wire and iron nails on a mudwat. Using my previous knowledge that the Geobacter and Schuanella species like to breathe iron compounds, I hypothesized that placing pieces of iron and copper into the soil would not only increase the population of the microbe species within the mudwat, but it would also increase the blinkage rate of it. In the perfect world of science measurements, I could have individually counted the population of microbes, but unfortunately I had to stick with observing the microwatt outage. I had 3 working mudwats, which I labeled mudwats A, B, and C. I kept mudwatt A as my constant, as it was giving out around 80 microwatts of power compared to the closer initial microwatt outage of 54 and 40 microwatts of mudwatts A and B, respectively. I cut the iron nails and copper wire into 1 centimeter pieces and put 5 grams of each into the mudwatts, with mudwatt A containing the copper and mudwatt B containing the iron. As you can see from the graph, the copper mudwatts microwatt outage continued to increase for a short period of time, but then rapidly began to decrease, dropping just around 12 microwatts in the span of 2 weeks. Mudwatt B, on the other hand, climbed steadily, increasing by 18 microwatts in the span of 2 weeks. I came to the conclusion that my hypothesis must be incorrect, as the copper mudwatt was decreasing. Mudwatt B with the iron had, on the other hand, been constantly increasing, which seemingly validated my hypothesis. I also observed several black spots that had formed on the walls of the mudwat, which you can see on the picture on the screen, and I theorized that the iron in the soil, while exposed to the water in the soil, oxidized and actually rusted into black rust. Black rust forms when iron 3 oxides with limited oxygen, like in the mudwat environment, and low moisture, which can result in thin black films or stains. The difference between black rust and red rust is that red rust, also called hematite, is the last and final step of iron oxidation, while black rust, called magnetite, is only partially oxidized and occurs prior to red rust. There are more electrons on the magnetite black rust, which means that it is more reduced. This also means that black rust could be a source of electrons into the microbial system, while red rust would most likely compete for electrons from the microbes themselves. Here is where the problem comes in. I had previously thought that the nails that I had used were already rusted because they had a thin film of like a kind of orangish brownish coating, but this was disproved as most likely only the topmost layer of the nail was rusted and not the other 90% of it inside. And so microbes love to eat rust, so it was essential that the entirety of the nail was rusted. After a discussion with the Mudwat team, my results finally made more sense. Copper is actually antimicrobial, meaning that not only does copper prevent cell respiration, but it actually destructs the microbe's DNA, which prevents it from reproducing. Microbes reproduce through binary fission or asexual reproduction, meaning that they grow at an exponential rate. This is quite the breakthrough realization as it deconstructed the entirety of my previous hypothesis. Inserting copper into the mudwat actually made it slow down. On the other hand, the data recorded from the iron mudwat did not align with the science behind it. The black rust should have slowed the mudwat down because it wasn't fully oxidized, but instead it increased the mudwat outage rapidly. The mudwat team and I theorized that there was some other form of unnoticed variable within the mudwat causing this change, as the iron could not be responsible for it. I think that this other variable could probably be mold, or maybe I added too much water, or there must be some other form of variable because it couldn't be the iron that was causing it to grow.
So hello again. So for my experiment, what I specifically wanted to analyze was the artificial and natural wavelengths of light, the pH levels, which can alter under certain conditions, and lastly, the fungi that grew due to the acidic environment. So for this experiment, I had to use an LED light bulb and some lemons that I just got from the store. And so what I found interesting about my experiment was seeing the mudwat have an increasing rate of growth under acidic conditions because methane is non-acidic and normally bacteria will grow depending on its pH environment. And so as you can see here during the, I believe it was on the 21st is when the 16th is when I started to look at the pH levels. You can see it going at an increasing rate later on. And so for my second and third variable, which was the mud I got from a dark natural light and from a sunny natural light, where I just like placed it outside because I was also looking at the alternating wavelengths. You can see it going at a faster rate later on in the experiment. However, when studying just like dark artificial light, which I use for an LED light bulb, I saw that under certain types of wavelengths, which is seen better at an orange or like red wavelength, it's able to, the electrobacteria was able to just like go at a faster rate at the very beginning, and it was able to stay at a constant rate. And so I made that the constant, and then later on I made the dark natural light and the sunny like natural light my two variables that I wanted to test out with different types of pH levels. And so what I saw is that acidophils, which is like types of, which is like what I thought this was going to be, which was the the dark natural light, which had lemons in it. It's usually supposed to optimally grow at near a 3.0, which is lemons were, which is at a 2.0. So I thought it might work. However, which is where we got the mud that we got it from, which is had a lot of methane in it. It's usually in a neutral environment. So that wouldn't be the case. So at least just like looking at this during the 2 weeks of this analyzation, I noticed that the pH can speed up the bacterial growth. However, I also noticed that when you input a liquid with a low pH under a natural or basic environment, it needs to be given that same substance frequently to keep up the bacterial pacing. And with the addition of the lemon seeds, which was added later to the sunny natural light container or mudwat, I was able to see that there might be a frequent growth as well that can contribute with the organisms either in a beneficial or non-beneficial way, and which can also just like overall affect the organisms. And so with these moderate changes in the pH, it modifies the ionization of the amino acid functional groups and disrupts hydrogen bonding, which in turn promotes changes in the folding of the molecule, promoting denaturation and destroying activity, which in this case was able to actually just increase it, which I was able to find kind of weird. So the optimum growth pH is the most favorable pH for the growth of an organism. However, we also need to take in scientific errors that were seen in this. And so examples of this was the surface areas that can limit the growth of the microwatts. The seeds might have caused it to shift as well. So it might have not just been the pH, but the organic molecules that I added later on. The air bubbles and the placement of the cathode and anode that can cause less oxygen to be exposed. And lastly, the very beginning of the experiment, I kind of made a mistake of placing the mud watts for the sunny natural light and the dark natural light in outside in 98-degree weather in the mud watts, and the mud watts were not hypermophils, in which are like hot loving electricity generating bacteria. So the bacteria might have gone fried. But moving on with my experiment, my electricity generating bacteria were bioavailable, meaning that it's easily broken down by microbes. For example, things that mold and rot are bioavailable, which was my independent variable, the lemons. And because of its acidity, which is a good energy source for these microbes in this case, we can see that it was able to grow at a good rate. However, if I were to put the lemon in the anode, then this will make the anode environment more acidic, which is bad for the microorganisms. And so lastly, with the fungi that was growing at the very end, it was able to outcompete abiotic reactions and ferment and create a lot of anoxic acids, which apparently was actually good for this environment. And so moving on to the next slide, you're able to see how in the dark natural light and for the sun natural light, how although it had some like moss or like fungi on it, it was still able to grow bacteria. But although I wasn't able to get like an actual good like video of like the actual bacteria like moving and things like that, it was able to grow. There was like, and it was at the very end that I kind of looked at it under a microscope. It was able to grow kind of like you're able to see some bacteria move. And looking at the dark artificial light, it was just always at a constant rate. And so bacteria was just always there, invisible and visible, sorry. So yeah, these are the images of the microorganisms that were inside each container of mud. And unfortunately, I couldn't get a video, as I said before, of like the organisms, but they're able to move slowly, which was pretty cool to see with our microscopes. And so yeah, taking under like these type of scientific errors and types of conditions and hypothesis, I was able to see that your hypothesis can definitely alternate or change during the given weeks with the given experiments that we were all able to do.
Yeah, so moving on to our general takeaways from this project, I found this experience to be extremely impactful on how I think as a scientist. Although I didn't go into this summer's program with much knowledge on microbiology to begin with, exposure to sometimes very dense reading material and repeated mistakes in my experiments have taught me that encountering obstacles in science is normal. Sometimes your results don't always turn out the way you want them to, and it can get kind of frustrating sometimes. But you just have to keep researching what may have happened and theorizing solutions to it. Dr. Newman always told us that good scientists do everything in their power to disprove their hypothesis if it's right. And that has taught me that making mistakes in your experiments is okay. In addition to this, we also had the privilege of reading lots of scientific articles about microbiology and actually meeting the authors of those same papers every Friday. I was very nervous at first, but Dr. Newman told us that these professors and researchers were normal people just like us. Meeting and talking to all these professors really changed my perspective of scientists at top universities, as I thought they would be so much more scary, but they weren't. Dr. Newman has also taught us to view microbes as not just pathological, but as helping more than harming us. Whether that be digesting food in our gut, purifying water, providing electricity, and more, microbes are essential in researching sustainable energy, water treatment and purification, and human health. For example, the picture here on the slide, the one at the bottom, is one of the first real-world microbe application examples that Dr. Newman showed us. It pictures a setup for an alternative natural water treatment method, which involves electroactive bacteria to purify the pollutants in the water, essentially making it safe for people to use. This is called bioremediation, using microbes to break down environmental pollutants and is incredibly useful for a sustainable future. Microbes are also potentially helpful in the fight against climate change, as many of them like to eat carbon. A fun fact is that a couple of years ago, a rust eating bacteria was actually discovered on the Titanic. It was fittingly named Halomonas titanicae, and although it is slowly eating away the Titanic, it could be very useful in recycling old iron ships and returning that iron to nature in the future. Overall, I am very grateful to have been able to experience this program, and I'm definitely going to be pursuing some form of a microbiology experiment on my own time in the future. Elijah, I think you're up next.
Some of the interesting and favorite parts of about the project are, of course, the experiments. But my favorite part personally was the speakers. Everything that they gave to us, all the knowledge that they gave to us on their respected fields, really opened my eyes about what we can do in science. It really made me see the bigger picture. As you can see, the bigger picture uh, is to fight global warming so that we can create a better environment for us. Uh, we were They were telling us how they are trying to create different microbial fuel cells such as us, so that um, there can be different ways to create energy. That's it.
Yeah, I agree with Elijah. My favorite part also was the guest speakers because being able to relate an experiment that we're conducting by ourselves at home to something that's happening in the real scientific community was super cool. Reading their papers and speaking with them on Fridays explained and reinforced the knowledge from the results that we were getting. So there were a lot of results that we were like, why is this happening? And just being able to talk with them and clarify any questions that we had was super helpful. It was a great experience to be able to relate something that we read in a paper, just something that was published in some sort of scientific magazine or journal, sorry, to something we were seeing happen right before our eyes. Reading these papers and meeting with the authors also helped me realize how systems like our mudwats can be used in the real world in larger scaled applications. There was a lot of talk about how devices like these can suck carbon out of the air and reduce the greenhouse gases in the air, so fighting climate change. And I can't wait for this to be used on such a large scale that we can actually combat climate change.
So yeah, in addition, I just also found it very interesting and I really love speaking, getting to ask questions and just like relate to a lot of things that the speakers were talking to us in the past Fridays. And I also just loved talking with my group every like Monday about our experiments and how they went, and just like asking Dr. Newman and Lev and Alex like a bunch of questions that we had about like our actual mudwats and seeing new ways and alternative ways that we can create like a new hypothesis or a new experiment as well. At least for the scientific papers, I love just like, again, just like talking with the authors as well as just like getting to like ask a bunch of questions and do a lot of like reading the papers and understanding them as well as a whole entire group. And then also just looking at the very big picture, seeing how these mudwat systems that we were able to create can be used for just like removal for many different environmental pollutions, including toxic heavy metals, uranium, that are contaminated soils and wastewater, and as well as just like overall looking at new renewable energy sources that are self-sustaining and do not require any external energy to operate, neither emitting any excess greenhouse gases, which is really good for the environment. And so at least in the future, I kind of want to look more into microorganisms because this was a field that I was really introduced to like this year. And so with the help with what we've been doing for these past weeks, which was an amazing experience, I want to look deeper and dive deeper into this topic and seeing how these microorganisms and different types of renewable energy sources could help our environment and help with like the emissions that are happening in our world.
And then just lastly, we'd all like to say a very special thank you to Dr. Newman, Lev, Alex, and Caltech for making all of this possible. They gave us the creative freedom to really pursue any experiment that best intrigued us while giving educational and informative advice on how to proceed. They also never failed to give us a fun fact or tell a funny story during our meetings, which I really appreciated. We could not have done it without all of you, so thank you very much.
That was so fantastic. I just have to say, you guys totally made my day. I really loved that presentation. And it's been just such a total delight to work with all of you. And I'm just so thrilled that this experience was meaningful for you guys. I have a few more remarks I'll make, but I'll wait, let everybody else ask some questions, but I just couldn't help but say that right away.
Any questions from the other students or anyone? Anyone's got a question? That was fantastic.
I'm
Thank you
gonna
all.
put something in the chat actually that is an attempt to try to link between the fields that we cover collectively, comparing the number of microbes to the number of stars in the universe.
Hey, this is Mike. Can you hear me?
Yes.
Ah, good. I wasn't sure if I was actually if this is working while we're on the road here. This was great. You guys did a fantastic job, and I'm jealous that it looked like you got to actually do some stuff in person. At least go collect the stuff on campus, which you know, we didn't get to go pick up our asteroids in person, which is too bad. But I'm really curious how the whole process worked. So did you guys all went together and collected stuff and then brought it home and chose experiments? Or so I guess my real question is how did you choose what to do, and how did it all start? It's just kind of all fascinating.
Well, as Maddie said earlier, we were given a lot of creative freedom when picking what we wanted to do. So we had a general understanding. Like in the beginning, we learned what is a mud watt, like what does it do, how does it function, like the basic stuff. And then after learning the basics, we were given the opportunity to pursue whatever path we wanted. So if we wanted to add cleaning products to see what that would do, or if we wanted to put our mud watts in different types of light, we were really given the option to do anything that we wanted to do.
So given the option is great. So then how did you each choose what to do? It's really hard and fun to get to do that. But so how did you choose?
Yeah, so we ended up, oh, sorry. We ended up coming up with a list of variables that affect a mud watt's performance. So things like temperature, light, etc. And then we came up with another list of things that could affect those variables. So then we would just choose from that second list.
That's great.
If I can just jump in a little bit to the earlier part of your question, the differences started from when we were first even collecting the mud, because, for example, the pond, part of it was in the shade and part of it was in the sun. So there were already temperature differences, illumination differences that the students noted as they were starting and choosing where to take their mud.
That's great. I mean, it just sounds like such a fun experience, but also a chance to really do some experiments and do some creative thinking and really learn a lot. It just sounds like a fantastic summer. Congratulations to you and to Diane and the rest of you people who were helping out. It was just great.
So I guess I have a couple questions about almost like the mechanics of the mud watt. Maybe this was kind of already covered, but in terms of turning on the light bulb, I assume, was that caused by like a potential difference between the anode and the cathode? Is that right? From the electrons, or how exactly does that work?
Say the question once more, please.
Okay, so basically, I'm just wondering how the, I guess, accumulation of electrons ultimately managed to maintain that, I guess, current through the light bulb, right? The LED, because, you know, just having electrons doesn't guarantee that they're going to move. And I would think that, I guess, there must be some mechanism of diffusing them at the top, right? Because otherwise they're just going to build up and then you're going to kind of lose the current eventually, right? So no, maybe one of you can kind of speak to that a bit.
So yeah, at least for like like experience, just like seeing how many alternative methods such as like oxygen and type of like the water that needs to be contributed. And so if you have like a dry mud watt or like just like no water at all, it can also just like alter like the electron like movement, because there's a lot of like types of I'm forgetting like the specific word, but there's a lot of not equations, but certain types of like not well, channels sort of that are involved in the actual mudwatt system. And so it needs like the contribution of like oxygen and like the air bubbles and like H2O that's like involved. And so if it's in a dry environment or if it's like in certain temperatures, that can alter that effect, causing it to decrease. Or if you have like sort of things like iron or like electron donors or acceptors in your actual mudwatt system, it can also make it increase as well. And so that's at least like what I got from like this like past experience, and like seeing how if you were to like just like have it too like too heated or just like have too little water, it can cause it to decrease extremely.
And then also to add on to that, the electrons would usually, or I guess the microbes themselves would usually cluster at the bottom, or I guess like at the top of the anode. So there's the cathode, the soil, and the anode. And then the microbes would be near the bottom or the top of the anode, and they would send their electrons to the anode. And then there's that wire that connects to the circuit board. And so the electrons would travel through there, turn on the light, and then it would travel back down to the cathode where it would interact with oxygen, and then it would produce water.
Everything you guys said is correct. I'll just make one small link to the question. The potentials are indeed different at the anode and the cathode. At the anode, you have a much lower potential than at the cathode. And it's that voltage difference that is driving the flow of the electrons, as you said. And so the electron acceptor at the cathode is ultimately oxygen, which has a very high potential. And by the time you go down in that little device, all the oxygen has been consumed by the microbes. And so the bottom of the device is anoxic. And the microbes are then able to utilize the organic carbon that is in the bottom of the device, oxidizing that. And as Maddie and Chize were just explaining, passing the electrons from the oxidation of that carbon to the anode. And that's happening at a lower potential. And then those electrons in turn through the circuit, like in a battery, wind up finding their way ultimately to oxygen at the cathode.
Okay. And then I guess the other thing was, I heard about the LED. I think maybe I might have missed another part, but how do you, I guess, actually measure the kind of wattage that you're getting through it?
So yeah, at least for like mine, I kind of just like I don't have like a super like fancy like LED light, but it just has like certain modes on it. And so for that, I was just able to I just put it at like I believe the second mode for like the LED light. And so, and then after that, there's different types of modes. So you can put it to like I believe like white, orange, or just like yellow sort of. And so I wanted to look at orange because it shows that usually at like or sorry, not yellow, blue. And so then after that, I wanted to see how the difference between like the orange and blue type of like light, since like I was trying to see kind of like orange if it's in like the natural or just like outside in the sun, or blue if it's like kind of in the ocean, and see if there's kind of like a difference between those two. And so for that, just like seeing like the difference, I just put it at like a mode or like level 2, and then just kind of like tested that for about like a week, and saw that usually at like an orange light, specifically because of like photosynthesis and sort of like those types of like reactions and things like that, it's able to work much smoothly under those conditions. But yeah, I just like have like a light bulb that I just like bought from like Amazon.
And then also, I actually did not do it that way, but we also had this app called the Mudwatt Explorer app, and so it has a lot of different features. It says Discover, which is like a little guidebook that features Shui, the Shuinella microbe. And then we have this thing called Measure, which is where it's a scanner. So you can actually point it. This is like the little box where you point it at, and you point it at the blinking light. And I'm not sure how it captures it exactly. Maybe it has like a rate at which it blinks per
I think it's
second or something like that. But it manages to record the microwatt outage, and then that is transferred to our data table, and you can have multiple microwatts at a time, and you can like record all the different outages that you have.
How long under optimal conditions would a mudwatt last? Like, does it only like live for a couple weeks, or can it live for like months or years?
Under optimal conditions, I'd say forever, because I guess it has everything it needs.
Yeah, like at least like since like the mud is full of like a lot of like complex sugars and things like that and other nutrients, and just like or if you're like able to use it from like where we got it from, it can like also just like accumulate or like last for like not like I wouldn't say like millions of years because I feel like I might be wrong, but it can last for like a lot of like years, or like I believe for like yeah months to years at least will be like a good like variety.
I guess as a you say kind of crazy or hypothetical, would one be able to make almost like a big mud watt with like an actual like chunk of soil where they had like this just massive anode wire up to like a cathode on top of soil? In which case then they have like a bit of oxygen near the top and maybe like rain for the water, and then the soil kind of replenishes itself on the standard cycles. Is that possible or no?
I think it is possible, especially if you think about like the real world application. Like we were studying the bacteria in Lake Oneida, or like, not the actual bacteria, but the bacteria in Lake Oneida. And if you think about it, that's kind of like a huge mudwatt where the sediment at the bottom of the lake is like the soil in our mudwatt. And just if you think about like the sediment in a body of water, it's like an anaerobic environment, but the same processes are still going on.
Yeah, I'll just chime in and say these great cycles of oxidation and reduction reactions that you capture in these cool little devices are basically what sustain all life on Earth on a planetary scale. And so, where you're getting the source of energy that you capture for any organism, it comes from somewhere and it's conserved. And so there are these redox reactions that recycle elements in different oxidation states, and depending upon what type of life form you are, you're able to tap in at different levels in that cycle. And so microbes are really the masters at this. They're the most gifted biochemists on the planet. We humans are very boring in comparison, but this type of energy conservation that you see here with these mudwatts is just a tiny example of the type of extraordinary energy conservation that microbes do in all different parts of the world, and including within our own bodies. The microbes in your gut are doing some pretty cool anaerobic metabolic processes that aren't that dissimilar from what's going on in these mudwatts.
Great questions, everyone. I'd love to hear from Alex, your perspective as the teacher helping lead the group, and how it might apply to your work in the classroom in the future. Any thoughts on that?
Oh, I have notes and notes of thoughts. I just keep jotting them all down. Yeah, everything has just been wonderful. What I just from the guest speakers, just and I've done a lot of summer programs, but this one is really something special, and it really, from the teacher end, from being kind of on the student end, it's just giving me a lot to take back into the classroom, and I'll definitely be using lots of stuff to take back. So it's been great. Those guest speakers every Friday just blew my mind, and I just it was amazing. Thank you, Diane, for bringing them into our summer. It's been really great. Thank you, Mitch. And students, you've been amazing. And I feel like this every time kids present. They exceed my expectations, and they just do so well because they're awesome. So great job, students. Chize, Caitlin, Maddie, Elijah. Awesome. Future scientists. Yay.
Yeah, thank you.
Thank you.
Anyone else? Any other parting thoughts? I'm so impressed with everything. As always, you guys did a great job, and it was just exciting to see that you were able to conduct these experiments and really think about how you were changing it rather than using a recipe or a menu. You know, you were actually just like, what can I do to change this? How can I make it different? What if I try this? What if I try this? So I would say not future scientists, but you are scientists already.
Yeah, I want to second that. I think you guys were so resourceful, and being able to do an experiment during this weird COVID time we're in at your home is actually not very easy. And what I really thought was so wonderful was just how creative all of you were, and how you totally embrace the scientific process, which is that you actually learn more when things don't go according to how you think they might go. That's when we actually learn the most. And that you had such an enthusiastic and really scholarly attitude, and how you would always go beyond what I gave you to read. You did your own research because you were curious. And I thought that was just phenomenal. It was a total delight to work with all of you. And I think you guys have exactly what it takes to be independent creative scientists. And I'm so excited to see what you wind up doing in the future. And I look forward to seeing you for a grand finale this weekend when we light the pond on fire. We're going to light the methane in the pond as a celebration of microbial diversity as our closing out.
Oh, wow. Wow. Maybe I don't want to know about that. I don't know.
Yeah, you didn't hear that. You didn't hear that.
It sounds wonderful. I'd love to see that.
Yeah.
Someone be running your camera when that happens.
Well, I also want to thank Lev. So Lev is a grad student in my lab who was a wonderful partner in helping me lead this group. And Alex, you were fantastic as well. So it was just a totally positive experience. So thanks to everybody.
So I think that concludes our Zoom meeting.
Yeah. Thank you all. Thanks.
Thank you,
Thanks, everyone. Thank you. And we'll see you on Sunday, everyone. Okay.
Bye.
Thank you.
Bye. Thank you. Bye.
Thank you.